We will do the surface epithelia today. So if you remember from the first lecture, then the epithelial tissue we can divide in main categories, and the most important maybe is the surface epithelium, which is found on the inner and outer surfaces of the body. Okay, other types are the glandular epithelium. Okay, I just misspelled it here. I will correct it. Myoepithelium, sensory pigment, and reticular epithelium. On Thursday, we will talk about these topics as well. So today we will discuss the surface epithelium, which are found on either inner surfaces of organs, of luminal organs, which have a lumen or a cavity inside, or they are found on outer surfaces, just like on the skin, the outer layer of the skin. We already began with this slide last week, so that there are two main types of surface epithelia, simple and stratified. The main difference is that in case of simple epithelium, each cell is in contact with the basement membrane. In case of the stratified epithelium, only the lower layer, the lowest layer, is contacting the basement membrane, and the upper layers are sitting on top of each other. And after we divided the surface epithelia in simple and stratified groups, then we can further divide them according to their shape. Okay, and according to the shape, we can have squamous epithelia, cuboidal epithelia, and columnar epithelia. And to be able to distinguish between these epithelia, it's always very important to be able to orientate yourselves in a slide. I just have a few examples here. These are just very small portions of different slides where you can see different types of epithelial cells. So first of all, what you have to do is to think in 3 dimensions, although you can see the pictures only in 2 dimensions. Okay, so if you see a structure like this on the right upper corner, okay, then you have to try to imagine this structure in 3D, okay, which can be difficult at the beginning. Okay, so this is in 2 dimensions. We can see an oval structure with a lumen inside. Okay, so if we try to transform it into 3 dimensions, then most probably this will be some kind of tube which we cross-cut. Okay, in the middle of the tube we can see the space, the lumen. Okay, and around the tube you can see some kind of other tissue, which is, in this case, it's a connective tissue. So if you see a structure like this, then you should know that where you could find the apical surface of the cells and where you would find the basal surface of these cells. Okay, the basal surface is always the opposite surface, opposite from the lumen. Okay, so the basal surface and the basal lamina would be here in this case, in the case of this given structure. Okay, so this is the basal surface, and the luminal surface would be here, the apical or luminal surface. Now, we can see mostly the nuclei of the cells. The nuclei are these basophilic, these dark blue structures in the middle. Okay, and the cytoplasm is eosinophilic, it's pinkish. Okay, the direct borders between the cells are not visible. We can just assume where the borders between the cells are. how can we assume it? We have to draw a line actually at the middle, or in the middle between two nuclei. Okay, and this is where the cell borders should be approximately. Okay, so here, here, and so on. Okay, so these are the cell borders approximately. And if you do this, then you will be able to tell if the cell is a squamous cell, a cuboidal cell, or a columnar cell. Okay, in case of squamous cells, the cells should be more wide than their height. Okay, the cells are wider. In case of a cuboidal cells, the width and the height of the cells are approximately the same. In case of columnar cells, the cells are much taller, much higher than their width. Okay, so if you take a look at this, then you can see that approximately it's the same. So a little bit taller, the cells are a little bit taller than their width, but still it's more like a cuboidal cell. Okay, in the lower right corner, if we do the same, this would be the base of the cells. This would be the apex, apical surface of the cells. And if we would draw the cell borders, then we will see much taller cells. Okay, these are columnar cells. So if the cells are elongated and they are perpendicular with the basement membrane, then they are columnar cells. Okay, usually the cuboidal cells have round nuclei. Okay, so this is again a big help for you. So if you see round nuclei, then most probably you are looking at a cuboidal cell. Okay, if the nuclei are a little bit more elongated, then these would be columnar cells. On the left upper corner, this is what you can see as well. It's also a columnar epithelium. Here the nuclei and the cells are also elongated. Okay, here the basal lamina, the basement membrane actually would be below. Just a moment. Okay, here would be the basement membrane. Here would be the apical surface towards the lumen. Again, this white space on the upper side, that would be the luminal side. And then we could draw again the borders approximately in between the cells. Okay, so these are again elongated cells, these are columnar cells. How would a simple or a squamous cell look like? This is what we can see on the lower left corner. Okay, this is a very small, small structure with a small lumen. Okay, again, towards the lumen you will have the luminal, the apical surface of the cells. Here you will have the basal surface. And then again, the borders you can draw or you can imagine as the halfway between the nuclei. Okay, and now what we can see is that the cells are wider than their height. Okay, and the nuclei should be actually a little bit more flat, but it also depends on the plane of the cut. Okay, so this is a more flattened nucleus. Here we can see a round nucleus, but if there is a nucleus which has a shape of a disc, we can make different cuts in different planes. We can make a cut through a disc where it would look round, and we can make a cut where it would look flattened. We can make a cut where it would look oval. So you should always think about these things. This is why In some cases, you can not only see flattened nuclei in case of a simple squamous epithelium, but sometimes it could be more round if we take a look from above, for example. Okay, so of course you don't have to draw this all the time. After a while, your eyes will get used to this, but this is how you could imagine actually the borders between the different cells, and this is how you can, this is the easiest way how you can determine if an epithelial cell is squamous, cuboidal, or columnar. Okay. Great. Now then let's go on. Let's see the simple squamous epithelia, first of all. In the case of the simple squamous epithelia, we can differentiate, we can distinguish three main types. We have the so-called real simple squamous epithelia, which has no specialties. This is what we can see in the first picture. And if you already had this week your histology labs, then you already saw this picture and this slide. This is from the papilla of the kidney, and here we can see different types of simple squamous epithelium. Okay, the first one is this real type. It's like the so-called Henle's loop in the kidney. Is this one, which we used as an example in the previous picture as well. Okay, so you can see flattened cells, flattened nuclei, and the cytoplasm of the cell is also visible, this eosinophilic cytoplasm around the nuclei. Okay, so this is a normal simple squamous epithelium, which can be found in the kidney or in the lungs, for example. Another type is called the endothelium. The endothelium is a special simple squamous epithelium which lines the surface, the inner surface of all of our vessels. They could be blood vessels or lymphatic vessels, it doesn't matter, and there is no exceptions. All of our vessels are lined with simple squamous epithelium called the endothelium. Okay, and this would be very important, and it is a very, very frequent exam question, so you can you can put an exclamation mark here because it would be asked in the exam as well, for sure, from almost everyone, to be able to tell what kind of structure lines the vessels. Okay, so this is a vessel here. This is a small vessel actually, and what we can see is or are the nuclei of the endothelial cells. We cannot see the lumen now. Okay, so this is a cross section of a tube of a vessel, but because the lumen is filled with blood, still, we cannot see the lumen. You can see this brownish fluid inside, that is the blood, and on the side you can see the endothelial cells. And here on this picture, actually, you can see the main difference between normal simple squamous epithelium and an endothelium. And this is actually the cytoplasm of the cells. Okay, while in the Henle's loop, the cytoplasm of the cell is very nicely visible. In case of the endothelial cells, the cytoplasm of the cells are usually not visible. It's a very thin cytoplasm. Okay, so oops, you can only imagine a line connecting the two nuclei with each other. Okay, so here you can see the nuclei. The nuclei are usually also flat, but they can protrude inside or towards the lumen a little bit. Okay, so these would be the nuclei of the two endothelial cells which build the wall of this given vessel. And there should be a very, very thin layer of cytoplasm. Of course, all cells have cytoplasm, but it's so thin that it's not really visible, so we can only imagine it. And again, then the borders between the cells would be somewhere here if you halve the distance between the two nuclei. Okay, so this is actually a small vessel which has only two endothelial cells in the wall, and this is how you can recognize such a structure. On this picture, actually, you can see more of it here on the left upper corner, for example, or here. Now it's easy to recognize these vessels because there is blood in the lumen. You will see later on such, or maybe you've already seen it in the first lab, that on some of the slides the blood is washed out and you can see only a white lumen inside. In this case, the Henle's loop and the blood vessels would look more alike. So actually then it would be important to know the difference between the epithelial cells, and that will be actually the help for you to be able to recognize them and to tell them apart. Okay. The third type of simple squamous epithelium is the so-called mesothelium. The mesothelium looks actually very, very similar to the normal, to the real simple squamous epithelial cells. This is what you can see on the right lower corner. Okay, here. The main characteristic of the mesothelial cells is that they build the so-called serous membranes of the body. The serous membranes of the body are these three, what I listed here, the pericardium, the pleura, and the peritoneum. These membranes will cover the heart, the lungs, and the abdominal cavity, respectively. Okay, and they are called mesothelial cells because they are derived from the mesoderm, from the middle germ layer of the embryo. You will learn it later on in the embryology lectures in this semester. Okay, so this is derived from the mesoderm. This is why it's called mesothelium. And here you will, in the labs, you will take a look at this slide, which I put here on the lower left corner. Okay, which is a bird's eye view of the peritoneum, actually. And we take a look from above of the cells. Okay, and this is a so-called silver nitrate impregnation, where the silver precipitate is found between the intercellular spaces, and this is why we can see the borders of the cells. Okay, so these are flat cells. Actually, it is the same as if we would take a look at this epithelium here from above. Okay, and this is how you can see the cells. So this is actually not a real slide. This is only a little portion of this very thin layer of peritoneum. It was mounted on a slide, and if you put it in the microscope, then the light gets through, and we are able to see the cells with a light microscope. Okay, so this is the simple squamous. These are the simple squamous epithelia. The simple cuboidal, as we mentioned it earlier, here approximately the cells are as wide as their height. Okay, and they have mostly round nuclei. Okay, and this is a very, very important characteristic. Okay, and if there is a round nucleus, if there is a spherical nucleus, a spherical structure, then it doesn't matter in which plane we make the cut. The cut will be round. Okay, so if there is a ball and you cut it, it doesn't matter where you cut the ball, you will get always a round shaped cut, only the size could be different. Okay, here we can see two examples. The first one I already showed you before. It's from the kidney, also from a tubule of the kidney. And this other one is a duct, actually, of a salivary gland. You will see it also later on this semester. Here, what I would like to mention in this picture on the left side is the importance of recognition of the artifacts in the slides. Okay, so here this white space is only an artifact. So there is, in the normal tissue, there is no space here. But as we prepare the slide, then the tissues can shrink. And because of the shrinkage, the different tissues usually get separated from each other, and a little space will appear between them. This is such a space. Again, at the beginning, it could be hard to tell if that space is originally there or it's just an artifact. But with time, you will get used to it, and your eyes will get used to it, and it will be actually easy to tell apart. So this is, again, a tubular organ with a lumen inside. The lumen is not that very well visible in this case. We can maybe suggest that this is a lumen. Okay, and here are the cells building the wall of this duct, which have also round nuclei. So this should be also a cuboidal epithelium. If you would draw the lines, the borders of the cells, then you would see that approximately they are the same size in their width and height. So they are cuboidal cells.
Excuse me.
Yes?
What was this white material that you said in the left picture?
This is a so-called artifact. Okay, it means that it is originally not there in the tissue. It is due to the preparation of the slide. So during the preparation of the slide, something happens with the slide. So like here, it shrinks. This is what we call, for example, an artifact. An artifact could be a little, I don't know, like dust particle on top of the slide or on top of the tissue, or some dye on the tissue. You will see lots of examples of this. It's not possible to prepare always a 100% clean slide, and there are different types of artifacts. You can Google it if you want. There are very nice pictures actually which you can take. Okay, so it's something which will be created during the preparation of the slide, and it's not a normal structure of the tissue.
Thank you.
You're welcome. Simple cuboidal epithelium could be found in the body either with no surface projection on top of the cells or with brush borders. Okay, so the cuboidal cells can have small microvilli on their top, or there could be such simple cuboidal epithelial cells which doesn't have any surface projections on the top. Okay, so about the surface projections, we talked last week, the microvilli, the kinocilia, and the sterocilia. So on a simple squamous epithelium, we never have any surface projection. On a simple cuboidal epithelium, we can have microvilli on the surface. But because they are so tiny, we usually won't be able to see them. Okay, but that's with this we will deal later on. In case of simple columnar epithelium, again, we can differentiate between different types having no surface projections, having kynocelia on the surface, or having microvilli on their apical surface. And in the simple columnar epithelial cells, this is what we already discussed, that the cells are much taller than their width. Okay, their nuclei are round, elongated, ellipsoid, elongated, and the longitudinal axis of the cells and of the nuclei is perpendicular to the basement membrane. Okay, so there is a 90-degree angle between the axis of the cells and the basement membrane. Okay, this is an example, this duct is an example of simple columnar epithelium with no surface projections. And for example, here in the second slide, which is from the jejunum, from the small intestine, we can see a simple columnar epithelium with a brush border. So it means that it has many, many microvilli on the surface. Okay, so we have again these elongated cells with oval nuclei, and on top of it we can see a very nice layer of microvilli. And if you remember from the first lecture, I told you that the microvilli are surrounded by a so-called glycocalyx, which is actually a mucus fluid around them. Okay, and this fluid is what is actually better visible, not the single microvilli next to each other. Okay, on this schematic drawing, you can see the microvilli next to each other, and the glycocalyx is not visible in this picture. Okay, and again about the planes of cut, which can be very, very different. So if there is a simple epithelium, there is a way to make such cut where a simple epithelium would look stratified, where the nuclei won't be visible in one row, but in many rows above each other, as if it would be a stratified epithelium. Okay, how is it possible? I try to demonstrate it here on the lower part. So if there is a simple epithelium and you make a cut like this, where in this plane where this black rectangle is, then this is what you would see if you would take a look from above. Okay, so if you take a look from above, then you would see lots of nuclei next to each other. Okay, as if it would be a stratified epithelium. And it is very, very important that if you get a slide, if you take a look at a slide, then you should not only focus on a small part of the slide, but you should check the whole slide, the whole length and whole width of the slide, of the picture. Okay, because if you only take a look at this part of this slide, then you wouldn't be able to tell what kind of epithelium it is. But if you can see nicer cuts, so cuts where really the shape of the cells are visible, because that's more like a longitudinal cut through, if we would cut through the epithelium here in this frontal plane, in this case, then we would see nicely the columnar shape of the cells. Okay, again, this will be easier with practice, but if you take a look at the whole slide always, and this is what you have to remember from this whole story, what I'm telling you, that it is very, very important to take a look at the whole slide always, not focusing only to one part. Okay. But in the labs, you will practice this, and you can, if you find such areas, then you can ask your your teachers to help you recognizing the different structures. Okay, I will skip this one because I would like to focus on the next type, where the simple columnar epithelium has kinocilium on the surface. Okay, last week we discussed that the kinocilia are anchored to the centrioles of the cells, which will be visible as a line, the line of the so-called basal bodies in the slide. And if you see such line towards the apical surface of the cells, then you can be sure that these cells have kinocilia on the surface. Okay, with a different kind of staining, you can see the line with different color. In this case, it's black. Okay, and it's a little bit harder to tell where the real borders of the cells are, but somewhere here would be the basement membrane. Okay, of course, where you can see this white space, that would be the apical surface, that would be the lumen. On the left side, this would be the same. Actually, this is the same tissue with different stainings, with hematoxylin eosin and then iron hematoxylin. And then you can see the line of the basal bodies as an eosinophilic line in case of the hematoxylin eosin staining, and as a black line in case of the iron hematoxylin staining. And above the line of the basal bodies, you can see the kinocilia of the cells. Okay, this is what you can find, for example, in the fallopian tube. Simple columnar epithelium. So this was the simple columnar epithelium, and now we will go to the pseudostratified columnar epithelium. The pseudostratified epithelia belong to the simple epithelia. Okay, so if you remember at the beginning, we divided the epithelium into simple and stratified. Somewhere between the two should be the pseudostratified epithelia, but because all the cells are in contact with the basement membrane, in case of a pseudostratified epithelium, this is why it belongs to the simple epithelia. Okay, so what happens here is that the height of the cells are different. Okay, there are taller cells and shorter cells. Only the taller cells will reach the apical surface. The shorter cells are usually cuboidal, having a round nucleus. So actually the nuclei of the shorter cells are found more actually closer to the basement membrane. And in another row, in another layer, you can see the nuclei of the columnar cells. And this is why we call it pseudostratified, because it looks like a stratified epithelium, but it's not. So it's somewhere halfway between a stratified and a not stratified epithelium. Okay, so it belongs to the simple epithelia. What you have to know is all the cells are in contact with the basement membrane, but not all the cells reach the apical surface. Important is that almost all pseudostratified epithelium have some kind of surface projection. And actually this will help you a lot in distinguishing between pseudostratified and stratified epithelia, because on a stratified epithelium we never have any surface projection. On a pseudostratified epithelium we always have some kind of surface projection. Okay, so if you take a look at the picture, then you can see that the nuclei are in many rows. Okay, so in different layers. So we don't see the borders of the cells. So we don't see that, okay, this cell reaches the apical surface, this other cell doesn't reach the apical surface. So we we cannot be completely sure of if this is a pseudostratified or stratified epithelium when we only take a look at the nuclei. But if you take a look at the surface and you see some kind of surface projection, then you should be or you could be sure that this is a pseudostratified epithelium and not a stratified epithelium. There are other ways to distinguish them as well, but first maybe this is the easiest one. In this kind of epithelium, you will always see a layer with round nuclei. Okay, so these shorter cells which do not reach the surface, these will always have round nuclei, and these are found directly on top of the basement membrane. Actually, these are the cells which serve as stem cells. They have a very, very high mitotic activity. They divide a lot and very frequently, and these will replace the cells above which already die. Okay, so these cells serve actually as a stem cell layer, and these are usually more like cuboidal and having a round nucleus. So pseudostratified columnar epithelium we have either with kynocelia. This is very, very characteristic for the respiratory system. Okay, for example, the trachea, but other parts of the respiratory system also have this kind of epithelium. And pseudostratified columnar epithelium can exist also with stereocilia. Okay, this is found in the duct of the epididymis in the male. Okay, again, what you can see is that there is a layer of round nuclei. These are the shorter cells. And there is, above this, there is a layer of oval nuclei. These are actually the taller cells which reach the apical surface. And of course, only these taller cells will have stereocilia on their surface. Okay, and now we got to the stratified epithelia. We do not have too much time, but it doesn't matter. The stratified epithelia is where not all the cells are in contact with the basement membrane. Okay, these definitions are very important. Again, I like to emphasize this. We like to ask actually lots of definitions in the exams. Definition of stratified epithelia, definition of simple epithelia, definition of fibers, and so on and so on. You will hear it a lot, not only in histology but in anatomy as well. Okay, so stratified epithelia, where the epithelial cells, not all the epithelial cells are in contact with the basement membrane, only the lowest layer, and then the other layers sit upon this basal layer. The different types of stratified epithelia could be distinguished when you take a look at the uppermost layer, and the shape of the uppermost layer will define the stratified epithelia. If the uppermost layer has columnar cells, then it's a stratified columnar epithelium. If it has squamous cells, then it would be a stratified squamous epithelium. And there is a third type called the transitional epithelium, where the uppermost cells could be either flat or cuboidal. This is a very special type of epithelium. Okay, for each of these types, you should be able to tell where they occur in the body. And this is also, of course, important for every other epithelial types as well. So for the simple epithelia, pseudostratified epithelia as well. Okay, and how they look like. Okay, so stratified columnar, the uppermost layer closer to the lumen has columnar cells, so oval nuclei. The other layers could be either cuboidal or columnar. You can see them here. Okay, but always the uppermost layer is always columnar. Okay, in case of a stratified squamous epithelium, we can differentiate between keratinized and non-keratinized epithelium. Here, actually, if there is a keratin layer on top, then it's a keratinized epithelium. This is found in the skin. And if there is no keratin layer on the top, then it is non-keratinized. The different characteristics you can see here, and you can hear about and learn about them in the labs as well, because now we won't have time to discuss it very, very detailed. But I thought that it would be more important for you to see how you can differentiate between the different types of cells, because this is what I see in the labs usually, that at the beginning it's very hard for the students to tell if it's a cuboidal epithelium, if it's a columnar epithelium, or a squamous epithelium. This is why I emphasize this now in the lecture. The rest I try to write down everything in details, so you will find the most important facts here, and of course in the books and in the labs as well. On Thursday, we will go on with the glandular epithelium, and we will talk a little bit about the other types of epithelia as well.